Have you ever wondered why sometimes you can resist that tempting slice of cake, while other times you just can't help but take a bite? Our motivation to eat is driven by a complex web of cells in the brain. Researchers at Scripps Research have now pinpointed a specific set of neurons that play a crucial role in this decision-making process. The parasubthalamic nucleus, or PSTN, is a small and relatively understudied region of the brain. Scientists have discovered that neurons in this area control when an animal decides to take its first bite of food. These neurons use signals from within the body and sensory information about the food to influence our eating behaviors. This discovery sheds light on the intricate mechanisms that govern our appetite and eating habits. In a study published in Molecular Psychiatry on July 4, 2024, Scripps Research scientists set out to manipulate a group of PSTN cells that become active during binge eating episodes. In our study, we used a technique that let us turn on cells in the mouse brain that were activated by a specific experience. In this case, binge eating, said Jeff Dunning, PhD, a staff scientist at Scripps Research and first author of the paper. By turning these cells on like a light switch, researchers observed the changes in the animal's eating and drinking behaviors. This innovative approach allowed the team to capture and analyze the specific ensemble of PSTN cells involved in these behaviors. The research team found that activating the ensemble of PSTN cells sensitive to binge eating drastically altered the behavior of mice. Hungry mice typically start eating quickly once food is available. However, when these PSTN cells were activated, the mice were much slower to begin eating and surprisingly faster to drink water. Our results tell us that this specific group of PSTN cells guide the early stages of hunger-driven decision-making, before eating actually occurs, said Dunning. The effect on water drinking, though counterintuitive, might be related to prandial thirst, the phenomenon where thirst is stimulated as soon as we start eating. By manipulating smaller sets of cells within the PSTN, the team identified which groups were responsible for delayed eating and accelerated drinking. They also discovered another group of PSTN cells that promoted the consumption of sweet foods. Altogether, these results reveal that PSTN neurons exert a complex combination of functions, said senior author Candace Contet, PhD, associate professor in the Department of Molecular Medicine at Scripps Research. The findings may have significant implications for understanding eating disorders, where individuals struggle with either too much or too little control over the initiation of feeding. The decision to take that first bite or wait longer could be influenced by the activity of these PSTN neurons. This research opens new avenues for developing treatments for eating disorders by targeting these specific brain cells. Beyond food and water, the researchers believe that similar mechanisms may be involved in the loss of control over the consumption of rewarding substances, such as drugs of abuse. The team is currently investigating this possibility, aiming to uncover whether the same PSTN neurons play a role in addiction. This ongoing research could provide valuable insights into the neural circuits that govern not only eating behaviors, but also other forms of compulsive consumption. By understanding the role of PSTN neurons in decision-making processes, scientists hope to develop new strategies for treating eating disorders and addiction, ultimately improving the quality of life for those affected by these conditions.